Ladies and gentlemen, welcome here live in Bucharest on Quotidiano TV today in Compass with Dr. Filippo D'Amico from the very famous San Raffaele Anesthesia and Intensive Care Unit. Dr. D'Amico, welcome to Compass TV. How are you doing?
Good evening. It's a pleasure to be here.
Thanks a lot for having accepted my invitation. Ladies and gentlemen, the Anesthesia and Intensive Care Unit of the IRCCS Ospedale San Raffaele in Milan is leader in Italy and worldwide for the treatment of critically ill patients. Our today's guest, Dr. D'Amico Filippo, has been involved in PNEI, epigenetic divulgation and research, and he worked as an emergency clinician. He also volunteered in Bergamo and authored the ItaliCO study during COVID-19. The reason why you are here today, Dr. D'Amico, is because of a paper which you've co-authored together with Professor Landoni called Recent Exposure to Smoking and COVID-19. Dr. D'Amico, please tell us what has motivated you, what has inspired you, and how did you come across that there might be a connection between smoking and COVID? None of us would say that smoking is healthy. We do not promote smoking. We just try to unlock some of the secrets behind COVID.
Yes, sure. In the early stage of pandemia, what was observed was that pathology affected people at different levels of intensity. There were people who were affected in a very important way, developing severe pathology. Other people developed only fever or were even asymptomatic. Again, what appeared was that not everyone was susceptible to COVID-19. So some people in the same family some become infected, others did not. So was very heterogeneous, the manifestation of COVID-19. What was the focus of the scientists at the time was to figure out what were the predisposing factors to develop a severe grade of COVID-19 or to protect people from COVID-19. Honestly, we thought that in people with severe COVID, we will find a higher incidence of smoker because smoking
Obviously,
is associated with pulmonary disease, and COVID-19 is prevalently pulmonary disease. To do this, we look at the population admitted to intensive care unit at the time. So we analyze all medication and medical history of these patients. Surprisingly, we found that only the 3% of patients were current smoker. This is inconsistent with the incidence of smoking in the general population, because in Italy, 14% of people are active smoker. We thought that was a bias due to the little sample size. So we analyzed the literature, all the publication of this field, and we found the same results. So the percentage of people admitted in intensive care unit was around 2 to 7% of people that smoking. So we analyze hospitalized in the ward, people with people in the general population was positive to SARS-CoV-2, and we found that in all publications the incidence of current smoking was very, very low.
But
so yeah.
So if I'm interrupting Dr. D'Amico, It's interesting that because also another very famous medical institution, the Institute Pasteur in Paris, made a similar finding, and also colleagues of you in Australia, Brazil, made also similar findings that the incidence among non-smokers is higher than among smokers. Were you in touch with your colleagues back then, or just after you started writing and looking closer to the figures, you got in contact with other colleagues who found a similar symptomatology?
No, sincerely, it was independent research.
Okay.
So everyone together, the focus of everyone is to show the characteristic of patients, and there was a collateral finding to found a low incidence of smoker in this population. So everyone want to show just the characteristic, not to correlate smoke and COVID. Was a collateral finding, and so we not act together. But then we did a meta-analysis of the literature. Is reanalyze the literature and the study with this outcome, and we put it together, and we found that the relative risk to be a smoker, to develop COVID in smoker, was two times lower in smokers respect to normal population, to non-smoker.
And how long, what was like your first reaction, or like, for instance, of Professor Landoni? How do you believe, how could he or you and your colleagues explain this different susceptibility of smokers and non-smokers?
Yeah.
If we now would start to dwell deeper into the topic.
Yeah, we tried to explain this correlation. It's not easy, and it doesn't completely demonstrate why the smoke is protective to COVID-19.
Sure.
One of the main hypotheses is that the effect of nicotine on acetylcholine receptor is protective to COVID-19. Acetylcholine is a neurotransmitter that acts on two types of receptor. One is nicotinics and one is muscarinics. So acetylcholine is not just an hormone. Is the first hormone discovered by human. It is involved in numerous mechanisms. For example, taste and smells are two of the characteristic symptoms of COVID-19. The action of nicotine on COVID-19 in nicotine receptor could be the protective mechanism of cigarette smoking on COVID. Nicotine is believed to result in downregulation of ACE receptors that are the door to enter in the body for SARS-CoV-2 virus. Another explanation is the downregulation on the inflammatory pathway. So cholinergic pathway is involved in anti-inflammatory downregulation, and we know that in severe patients what happened is the cytokine storm, so over-responsiveness of inflammation in the body, and maybe nicotine can act downregulate this response. Moreover, we try to explain in another way. We demonstrate that COVID-19 have a seasonal pattern. We demonstrate that in temperate country, the incidence of COVID-19 was lower in the summer. So we try to correlate the heat effect with on SARS-CoV-2. We know that at 50 degrees the virus is destructed, and maybe the heat of smoke in the upper respiratory way can destroy the virus. We don't know which one is the main reason of the protective effect of COVID. We just know that there is a relationship with the smoke and the lower incidence of COVID-19.
Yeah, Professor De Bernardis is a clinical pharmacologist at the addiction service CERT Lenti in Syracuse at the province health agency, together with Dr. Bursa, and they published in July 2020 a paper, a putative role for the Tobacco Mosaic Virus in smokers' resistance to COVID-19. So he suggests a different pathway which might induce protection for COVID smokers. And I've interviewed recently Professor Robert Friedland from the Louisville University. He's a professor for neurology. And he published a paper about 10 or 11 years ago called Why Humans Produce Antibodies Against the Tobacco Mosaic Virus. And the reason why humans produce antibodies against a plant pathogen, which according to a canonic science cannot infect humans, is because of proteinaceous mimicries. So because of a coat protein of the tobacco mosaic virus, certain peptides are just similar with the Tom complex of the mitochondrial membranes. Therefore, there's a cross-reactivity, and humans start produce exactly the type of IgG antibodies which are protective against Parkinson and autoimmune encephalitis. So I'm not a physician, but since I prepared myself for this interview recently, I still have this in my mind. But that would mean that there must have been an exposure to the tobacco mosaic virus, and there are several studies, for instance, that smokers are in touch with the tobacco virus because about 40% of the cigarettes contain residues of the tobacco virus, but by burning the cigarette, this virus gets killed. So obviously by saliva or by touching the cigarettes, there might be a contact able to induce an immunity. What do you think about the thesis of Professor Bernardis and Busa?
Yeah, there is a profound interaction among the microorganisms of our body. We know that between 500 and 10 million different species of microorganisms are found in human, and yeah, there is bacteria, but there is virus or mycetes, and we know that each individuals have her own microbiota. The total number of genes in microbiota are 100 times number of genes in the human genome. The function of human microbiota is really amazing. If we study the microbiota, we found an interaction with hormone production. We found a function relative to degrades product to synthesize molecules. We know that the intestinal microbiota can act together to counterbalance the infection. So we can consider the microbiota as a metabolic organ that perform a function that we are unable to perform otherwise. This function includes the ability to assimilate components or fight against other microorganisms. this is maybe the key of lecture of the Tobacco Mosaic Virus. So if we know that Tobacco Mosaic Virus is not in fact human, but if we enter in contact with it, we can develop antibodies against, and some studies show the presence of these antibodies in humans, and further studies are needed to demonstrate it, but some studies that are emerging show that intestinal microbiota of COVID-19 patients is really different to general population, and some study focus on the presence of Tobacco Mosaic Virus in this microbiota. So we don't know really which one is the connection to Tobacco Mosaic Virus and COVID-19, but we can imagine that one possible explication can be this one.
Yeah, surprisingly, what you're saying about the microbiota, Chinese scientists from the Beijing Key Laboratory for Precision Medicine of Chronic Heart Failure and the Medical Innovation Research Division of Chinese PLA General Hospital in Beijing, in a paper published on COVID, obviously, also back to 2020, called The Active Lung Microbiota Landscape of COVID-19 Patients, they say that they found in all alveolar fluids of COVID infective patients, they found COVID-19, but in none of the lungs of patients infected with other respiratory viruses. Now, this is making the situation even more tricky. I understand that that the tobacco mosaic virus, let's say, may be on a tomato, which by eating the tomato, it might enter the digestive tract of humans. But there seems to be a difference in case of the lungs, that only in cases of COVID, they encountered the tobacco mosaic virus, but not in case, as they said, of other respiratory viruses. So as if they too would belong at a certain extent together.
Well, theoretically, microbiota not acts only with direct interaction, but indirectly. So we can have positive microbiota that act synthesizing or enhancing anti-inflammatory pathway, and microbiota that acting promoting inflammation. So we can have the same effect, the same anti-inflammatory effect if we modify the microbiota in gut or in the lung is completely different. So this could be the reason. We don't know where we have to consider that one Italian study analyzing the gut microbiota in children found that the flora, the bacterial flora, was really modified to an inflammatory pathway, and the composition of the flora act all together in an inflammatory pathway, so enhancing the inflammation and can produce this cytokine storm. We have to consider that gut is like is more than 10 meters.
It's like a universe.
Yeah. So we have to consider all together, not just the single point, the single changing, but there is really a population inside us. Okay.
Yeah, but as I said, the Chinese did not refer to the gut. They were looking to the lungs only, and I'd like to quote from the article. They say literally the abundance of 18 viruses significantly differed between the two groups. Actually, the healthy and unhealthy group, absolute value, in absolute value. In addition to severe acute respiratory syndrome related coronavirus, surprisingly, 13 of 18 differential viruses were only detected in COVID-19 group, such as pangolin coronavirus and Tobacco Mosaic Virus, TMV, that's the abbreviation, which were probably closely related to the infection of SARS-CoV-2, though the function of them is unclear. So they came a step, not necessarily further, but they investigated a different aspect, but they didn't come further either.
Yes, sure. We don't know how they act, as you said, but we know that there is a correlation between this finding. So it is impossible to think that for who do research know that everyone has had a meaning, and everyone can everything can modify the homeostasis of an organism. So when we think at microbiota, we should think to a little word that act together and that relays stimulus or degradate component, and the equilibrium, the homeostasis of this little world have to be considered, and introduce or remove a single family of virus, of bacteria, can change all in the homeostasis of this little world.
Yeah, I like, because it's also a very useful scaffold for the discussion. I'm always referring to several papers, also for having, as I said, a proper scaffold and structure in a discussion. Another extremely interesting paper has been also published by Chinese scientists back to 2012, and it's called The Invasion of Tobacco Mosaic Virus RNA Induces Endoplasmic Reticulum Stress-Related Autophagy in HeLa cells. Well, this autophagy, ladies and gentlemen, is a cell internal mechanism to decompose, to destroy a virus. Is that correct, what I'm saying, the way I've shortened what the role is of autophagy? Dr. D'Amico.
Yes, the study demonstrates that Tobacco Mosaic Virus can stimulate the endogenous lysosomal cells to degrade virus. Is easy to think about it because more of the signal of each virus are very similar. Viruses are structured more ancient in our evolution. So yeah, they have similar pathway, similar protein that express. So two viruses that are very similar can stimulate in the same way the endogenous system. So this China study demonstrates that the presence of Tobacco Mosaic Virus can enhance the degradation system to remove COVID-19 virus.
Yeah, to quote again, they said the Tobacco Mosaic Virus can start this autophagy, and then this autophagosomes, these vesicles, for instance, which get built by the Tobacco Mosaic Virus in this process of autophagy, are incomplete ones since they cannot be processed completely. And then they say that piconaviruses, and piconaviruses, it's like the polio virus, for instance, coronaviruses, and SARS, and one arterivirus, which is a horse disease, they replicate on the surface of the autophagosomal membranes. So my question is, if the tobacco mosaic virus may induce this autophagy, and on the membrane of these autophagosomal membranes, on the surface of these autophagosomal membranes, SARS and polio can replicate. If I was to be, let's say, a lawyer, I would say, from a legal point of view, it's a case of complicity. We cannot say it's a case of co-infection because the tobacco mosaic virus cannot replicate in humans, but it may trigger an immune process which might look like creating a breeding ground, for instance, let's call it breeding ground for SARS and polio.
Yes, sure. This is one of the main mechanisms of we use the microbiota in general in lungs, in gut, or everywhere. We use the microbiota for its synergic effect. So in general, our body or our inflammatory organ can memorize, can act. One of the main mechanism of action of this component is the memory or the action for similarity. So this is one of the main mechanism and the most important that we can stimulate, because we can use our body can use this known antagonist, this known enemy, to respond to other one new and with the same mechanism.
But what it would surprise me, I mean, there's a whole debate related to the origin of COVID. Is it from a bat? Is it from the pangolin? Is it from a cave? Or is it from a cage? Okay. But in case this mechanism would make sense that the replication of SARS and polio takes place on the membranes of the autophagosomes, then the tobacco mosaic virus, which is not part of our microbiota, is the definitely controllable part, more controllable part in this tango. It takes two to tango. The tobacco mosaic virus does not enter our blood. There are papers, there is lots of research, for instance, for inoculation mice and rabbits with the tobacco mosaic virus, and it needs a mechanical lesion. You cannot inhale the tobacco mosaic virus. It won't do anything. Smokers get in touch with it by touching it, but this does not enter the body. By smoking, the virus is destroyed. So there, in plant pathology, in phytology, the tobacco mosaic virus needs a little scratch. Ever since it's been known that it needs a mechanical lesion to scratch something, a surface, in order to have it proliferating the body.
Yeah.
No idea what might have, for instance, in case if it was such a tango, no idea what might have be the scratch of a mechanical lesion to have human beings getting in touch with the tobacco mosaic virus, because there's no other way they can enter the lung. The Professor Lomonosov, he's one of the worldwide most famous experts in the tobacco mosaic virus. He said by smoking it, he said like that, the virus gets killed. So there must be another mechanism how this virus would have got into the lungs of COVID patients and have altered the microbiota of the alveolar fluids. So I think this would be then the next question to investigate. How did this tobacco mosaic virus enter the microbiota of the lungs.
Yes, but maybe just get in touch with tobacco can and bring to the upper respiratory way. Not smoking can introduce this virus to patients. It is not necessary. Sure, smoking tobacco. It's impossible that tobacco induce the presence of this virus in our microbiota, because smoke destroy completely, is like 180 degrees, so it's impossible that some virus can resist to this temperature. But
yeah,
but touching the tobacco and bring to our mouths maybe is one of the way to mechanically transfer this virus from tobacco to our body.
That's what I have read so far, and I'd like to encourage all our listeners to really to read and look at this matter, particularly those of you who are familiar with, let's say, biochemistry, physiology, and of course medicine, because there needs a mechanical lesion. Touching is not a lesion. Now, I'd like to give you another example, which really seems to be very interesting. Lots of neurologists, and for sure colleagues of you as well, you've heard that they do compare symptoms of COVID-19 with encephalitis lethargica.
Okay.
Which is a very complex matter. The etiology of encephalitis lethargica is of unknown origin. It's one of the biggest mysteries in modern medicine. Now, surprisingly, Professor McNulty in the 30s, he described that, according to his view, there must be a sort of lesion of the nasal vault or the throat, which proliferates encephalitis lethargica. Back to 1919, Professor Bassot published a paper about the encephalitis lethargica and said that, in his view, it must be a sort of co-infection. Now, I can only draw some conclusions. I'm not a physician. I'm a simple moderator.
So
neurologists compare COVID to encephalitis lethargica. A very famous professor, like McNulty, spoke about the necessity of a lesion of the epithelial cells in the nose, and then the idea of a co-infection. No, in English you say what walks like a duck, and flies like a duck must be a duck. No idea how the tobacco mosaic virus, this I'll have to investigate further, enters our body. But in Professor Friedland, for instance, explains, and also Professor Young wrote a very interesting paper about the immunological paradox. By touching peptides of the coat protein of the tobacco mosaic virus induces a sort of immunity which protects the glutamic receptors against an inflammatory process. So it's not the entire virus necessary, because the entire virus may create lesions and may induce an autoimmune reaction.
Okay. Lethargic encephalitis is really a pathology that affects in the first 50 years of 19th. It was really a pathology that was correlated with Spanish flu because timing. So there was Spanish flu in the first 20 years, and later became this encephalitis. The etiologism of this pathology was never been detect. So we don't know what's happened. At one time, disappeared completely. So all physicians of that time thought that was associated with a virus. We don't know which kind of virus, but maybe a virus, because the lesion was similar to encephalitis, but we never found an etiology. COVID-19 is different. COVID-19, when we talk about long COVID, we talk about symptoms that affect people for more than 2 months, and after more than 3 months, this is the definition of World Health Organization, more than 3 months after COVID-19 infection. What can be in this definition is all. We can have pulmonary symptoms, or neurological symptom, or psychiatric symptom. In this area of psychiatric and neurological symptom, we can have I talk as a physician, of a clinician that study the pathology with symptom and clinic of patients. We have a spectrum of symptoms completely different to encephalitis, to lethargic encephalitis. The symptom of lethargic encephalitis were catatonic symptom. People cannot moving or talking, and was treated by anti-Parkinsonian drugs. And this was the reason why was associated with the alteration of dopaminergic pathway. In COVID-19, we don't have this kind of symptom, but we know that there are some substrates that are similar in this neurodegenerative disorder and COVID-19. Essentially, one of the main association is with dopamine and cholinergic pathway, and this is the same alteration that we can find in lethargic encephalitis, or in viral encephalitis and COVID. It's really impressive that what did you say before, and it's true that Tobacco Mosaic Virus is associated with this protective protection.
Yeah, Professor Karl Lauterbach, he is the current minister, the German current minister for public health. He tweeted, ladies and gentlemen, 2020. You are welcome to check what he posted on the 21st of July, 2021. I'll have to translate, since he wrote it in German, that in the 20s and 30s, there was a worldwide wave of encephalitis lethargica, which was due to the Spanish fly. This is maybe not the case due to, but let's say it was due to the Spanish flu, not fly, flu. The brain lesions appeared years after the flu. This is unfortunately also in case of 19 not to be excluded. If there is a causality between Spanish flu and encephalitis lethargica, this is a different kettle of fish. But what also Professor Foley, a famous Australian researcher on EL, said that it took experts several years to investigate encephalitis lethargica because symptoms sometimes changed. They were not identical in all patients. It was almost impossible for a scientist to investigate the disease for 10 to 15 years. So let's say 3 years after the pandemic, we are not at the onset, but still in a very early phase in order to say, do we cover yet all of the 29 or 30 symptoms of encephalitis lethargica, or just a part of them?
Yeah, we really don't know what will happen in the next year. We are in the early phase of pandemic, so we just pass the infection, the pandemic wave, but we don't know how the body will respond to the infection, the precedent infection, especially in that people that develop a severe disease. Spanish flu and lethargic encephalitis don't affect all people that had Spanish flu, but some people, and we don't know the characteristic of that people. So we don't know if a little population that developed COVID-19 will have a neurological disease after this pandemic. This is impossible to know, but we have to take in consideration. We have to learn from the present pandemic wave that was very similar in the course, in the wave, in the timing, in the first of the century, and we have a lot of assonance.
Surprisingly, one of the striking similarities, experts back then, there were also lockdowns. And they've observed that closing schools made no sense because young children did not get encephalitis lethargica. And also in families, not all family members got sick. So they were aware of the fact that this must be a very unusual virus. Now, another interesting aspect is that experts in the Tobacco Mosaic Virus say that this virus has an amyloid structure and that it is the only virus to have an amyloid structure. Now, if I'm mistaken, please correct me. I read another paper written by Professor Mark Zabel, who explains very well prion pathogenesis. And I understand that dendritic cells are relevant for transporting the antigen further to B and T cells. Now, children don't have well-developed dendritic cells yet. There are also several studies related to COVID and prion disease. Could the lower susceptibility of children be related, in case we are talking about a prion protein folding process, could be that related to unmature dendritic cells, in your view, according to your point of view?
The neurological signs of COVID-19 are secondary to either inflammatory response. So we don't know if there is a virus tropism to the central nervous system. We just, especially in the first pandemic wave, was really impossible to perform a systematic analysis of central nervous system of patients who died, or just neurological analysis, clinical analysis of all patients, and reporting it, compare it. So what we know about the neurological aspect of COVID-19, we are learn today at this moment, not when there was the big problem of epidemic, of pandemic. What we know theoretically is that the main cell that regulate inflammatory response in in central nervous system is the dendritic cell. So the neuroinflammation, that enhance the neurological dam in prion or in lethargic encephalitis, that was the mechanism. The etiology was different, but the mechanism to develop neurological aspect of viral encephalitis is the reason, because we talk about encephalitic. Not encephalitic is a term, is a definition of an infection, but we have the sign of infection, but we don't have the etiology. But we talk about encephalitis because we see the aspect of neuroinflammation inside the brain, and what regulates this neuroinflammation is dendritic cells. So it's really is an evidence that there is a correlation between dendritic cells and neuroinflammation and the neuroplasticity of brain of children. We have to put it all together, it's sure.
Dr. D'Amico, thank you so much for this remarkably interesting interview with you. Thank you for taking your time, and wishing you further lots of success in your research. And I have the confidence that with what you came across, you really, and Professor Landoni have a point in that, and that you have a great potential to unlock actually some of the secrets of COVID.
Okay.
Thanks for having joined us. All the best.
Thank you.
And
Thank you.
have a nice afternoon. Bye-bye.
Bye.